And the next speaker is Amir Sapir from Haifa University, and he will send nematodes to space for us. Please.
Hello, everybody. I would like to talk today about nematodes as a platform for understanding fundamental aspects of animal life in extreme environments. And here is the best definition I found of extreme environments in the literature. And as you probably know, Earth is rich in extreme environments, for example, the deep sea, different kinds of deserts, and lakes. Here is Monolake. And these environments can be a model for extreme environments in other planets. And there is additional extreme environment which is interesting. It lies in the interface between Earth extreme environments and the planets, and this is the International Space Station, the ISS. So what nematodes has to do with all these extreme environments? So nematodes are probably the most abundant animals on Earth. They're typically 1 millimeter long invertebrates. Here you can see their position in the animal tree. And a common thread of many different extreme environments is the dominancy of nematodes. Here you can see 4 extreme environments, including our intestine, that are dominated by nematodes in comparison to other species. So our aim was to study extremophilic nematodes to understand how animals were adapted to thrive in extreme environments. And I will focus on 3 such environments: the deep sea, toxic lake, and the ISS. So let's start with the deep sea. The deep sea is Earth's largest but less explored environment, and specific habitats in the deep sea are rich in biomass but relatively poor in oxygen. I'm sure you're familiar with hydrothermal vents, and some of you are familiar with cold methane seeps. And what is interesting is that these habitats supports a very complex ecosystems. Here, for example, you can see bacterial mats and tapeworms. And we wonder how nematodes thrive in habitats relatively poor in oxygen. It's not only that these habitats are poor in oxygen, they're also rich in hydrogen sulfide, which is a potent inhibitor of respiration. And our hypothesis, quite naively, was that nematode adaptation to these habitats involved the loss of mitochondria and the evolution of anaerobic respiration. So to test this hypothesis, we collaborated with Victoria Orphan from Caltech, who studied hydrothermal vent and cold methane seeps in the Pacific Ocean floor, and we focused on one site, Hydroid Ridge, off the shores of Oregon. This is about 1 kilometer deep. Here you can see images of the site, the rocks, the bacterial mats, and it's hard to see, but actually there is a nematode here eating the bacteria. And we found that nematodes are the most abundant animals at Hydroid Ridge, and this is very consistent with samples from other sites, from other vents and seeps. And here are images of 2 of the species of worms that are living there, and you can see their interaction with microorganisms that cover their skin. And this is basically my favorite nematode ever. Look how beautiful this animal is. And to test our hypothesis, we took the nematodes and scanned them with transmission electron microscopy. Here you can see a single muscle cell of these nematodes, and you can see a nucleus and organelle that looks like mitochondria. So our initial hypothesis basically was not true. It's still possible that these organelles function anaerobically, and I will discuss that shortly. But while doing all this analysis, we found something quite strange and peculiar. It seems that the nematodes were eaten by some kind of an organism. And here you can see these are the body of the nematodes and the muscles, and there are spores and some other structures inside that are eating the worms. So we didn't know what this organism is, and after a lot of characterization, we end up with a parasitic fungus. This is Microsporidium. And Microsporidium does not have mitochondria. So we went for nematodes that do not have mitochondria, and we end up with a fungus that it does not have mitochondria. And it's interesting because these type of parasitic fungus is in some sense similar to viruses. They complete all their life cycle inside the cells of the host and spread through spores or some other forms that are metabolically inactive, and these spores inject the DNA of the fungus to the host cells. And this finding was pretty surprising because there was no report about microsporidia in deep sea ecosystems. So we wish to suggest that there is a hidden world of parasitic fungi in the deep sea, and that deep sea ecosystems are more complex than previously thought, comprised not only of bacteria and archaea and the organism that feeds on them, but also of animal parasites like microsporidia. And we wonder, is the anaerobic nature of microsporidia an advantage in habitats of low concentration of oxygen? And we wanted to continue with the microsporidia studies and with the nematode studies, what is the function of the mitochondria, but we had a problem. And the problem is that these nematodes were not culturable in the lab. And a second problem, it's really expensive to send up expeditions to the deep sea. So we decided to switch to a more accessible extreme environment, and this is Monolake. And this is a fascinating environment. It's a hypersaline, alkali, and arsenic-rich environment. And this is quite interesting because arsenic, like hydrogen sulfide that is fairly abundant at the deep sea, is a very potent inhibitor of respiration. So we went to the lake and sampled the lake. And immediately we found many different species of nematode. Here you see only 8, but actually there are many more. You can see here the images of some of the nematodes. So this is the first report of nematodes in Monolake, and actually we found that in terms of number of species, animal species, nematodes dominate the lake. And we're really happy with another fact, is the fact that we can isolate a single species of nematode, Anonema, that is culturable in the lab. So once we had a stable culture of monolake nematodes in the lab, we could ask more mechanistic questions about the adaptations of animals to extreme environments. And of course, the first question we want to address is whether monolake nematodes are arsenic resistant, and this indeed is the case. You can see here in blue is monolake nematode. In brown is the reference C. elegans nematode. And you can see here the quite dramatic resistance of monolake nematodes to arsenic, especially to arsenic 5. We wonder about the molecular mechanism of this resistance, and for that we sequence a gene, DBT1, that was shown previously to confer some of the resistance of nematodes and human cells in culture to arsenic. And we found that nematode from monolake and related species has a polymorphism in one residue of the protein from C to S. And you can see that this polymorphism probably is part of the mechanism that confers this resistance. Interestingly, there is a sister species to C. elegans that also has this polymorphism, and you can see that it lies in the intermediate between the monolake worms and C. elegans. So we believe that this polymorphism confers some of the resistance, but there are other mechanisms of resistance. So we propose that DBT1 polymorphism is one of the mechanisms supporting respiration in arsenic-rich habitats. So now let's switch to the ISS. So here our research aim was a little bit different. We wanted to understand how life in the ISS affects the nervous system in health and disease. And of course, this is important in cases of long-term expedition in space. So the approach was to use nematodes, the reference nematodes, C. elegans, that express a disease-prone protein, in this case Huntington disease-related protein, at their muscles. And here you can see the progression of the disease, of the Huntington disease, as manifested by the formation of aggregates in the worms. Very simple experimental setting. It's tubes filled with nematode at the central compartment at the dormant stage, food in one compartment and fixative in the other. So at space and in lab, the astronaut will open one gate to mix the food with the nematode to elicit their growth for 2 days, and then the opening of the second gate will fix the worms. So this was done in the ISS and in Earth. And as you can see, there is a difference in the pattern of aggregation of the Huntington protein, but of course you can easily see that there is also a dramatic difference in the shape of the worm in the developmental stage. So we still need, so this, we consider that as a preliminary experiment, and we still need to tackle the system and to make it more stable. And this is our future plans. But it was really important for me to show you this to give you a taste of the potential nematodes, and specifically C. elegans, has as a simple and very effective model organism for studies of different aspects of animal biology in space, using a very simple system like this. So with that, I would like to thank lab members and funding. Okay.